Hey there, science fans! Today we're diving into the fascinating world of cellular energy production. Hold on to your lab coats because we're about to explore oxidative phosphorylation, the powerhouse process that keeps our cells running. Think of oxidative phosphorylation as the grand finale of cellular respiration. It's the stage where the majority of ATP, the cell's primary energy currency, is generated. Without ATP, our cells wouldn't be able to perform even the simplest tasks. From muscle contractions to nerve impulses, ATP is the fuel that makes it all happen. Oxidative phosphorylation is like a complex, well-oiled machine, carefully extracting energy from our food and converting it into a form our cells can use. So, buckle up, science enthusiasts. Let's break down this essential process and unravel the secrets of how our cells stay energized. Imagine a relay race where electrons are the star athletes. That's essentially what's happening in the electron transport chain, a crucial part of oxidative phosphorylation. Embedded within the inner membrane of the mitochondria, the electron transport chain is a series of protein complexes like a chain of runners passing a baton. These protein complexes accept electrons from NADH and FADH2, molecules carrying high-energy electrons derived from the breakdown of food. As electrons are passed from one complex to another, they lose a bit of energy along the way. This released energy isn't wasted. Instead, it's used to pump protons, positively charged hydrogen ions, across the inner mitochondrial membrane, from the matrix to the inner membrane space. This pumping action is vital because it creates a proton gradient, a difference in proton concentration across the membrane. Think of it like a dam holding back water. The proton gradient stores potential energy, which will be unleashed later to drive ATP synthesis. The electron transport chain is a master of energy conversion, setting the stage for the final step in ATP production. Let's break down oxidative phosphorylation into manageable steps. First, remember the electron transport chain, our electron relay race. This process generates a proton gradient across the inner mitochondrial membrane. Second, we have chemiosmosis. This fancy term describes how the potential energy stored in the proton gradient is used to drive ATP synthesis. Think of it as harnessing the power of water flowing downhill. Third, there's ATP synthase, a remarkable enzyme that acts like a turbine. As protons flow back across the membrane through ATP synthase, they cause it to spin, providing the mechanical energy needed to convert ADP, adenosine diphosphate, and inorganic phosphate into ATP. Finally, the newly synthesized ATP is transported out of the mitochondria and into the cytoplasm, where it can be used by the cell for various energy-demanding processes. Section 4. Chemiosmosis, the driving force of ATP production. Chemiosmosis might sound intimidating, but it's a pretty straightforward concept. Picture this. We have a high concentration of protons in the intermembrane space, thanks to the electron transport chain. Now, these protons want to move down their concentration gradient from an area of high concentration to an area of low concentration, back into the mitochondrial matrix. However, the inner mitochondrial membrane is like a tightly sealed border patrol, preventing protons from simply diffusing across. The only way for protons to cross is through a special channel protein called ATP synthase. As protons flow through ATP synthase, they release energy like water spinning a water wheel. This energy is harnessed by ATP synthase to convert ADP and inorganic phosphate into ATP, the cell's energy currency. Chemiosmosis is a brilliant example of how cells leverage gradients to perform work. It's like a natural battery, storing potential energy and releasing it when needed to power cellular activities. Section 5. ATP synthesis, from protons to energy currency. ATP synthase is the molecular machine responsible for the final step of oxidative phosphorylation synthesizing ATP. This remarkable enzyme is embedded in the inner mitochondrial membrane, with a portion extending into the matrix. Think of ATP synthase as a rotary motor. As protons flow through a channel within the enzyme, they cause a part of the enzyme to rotate. This rotation drives a conformational change in the catalytic head of ATP synthase, located in the matrix. This conformational change provides the energy needed to bring ADP and inorganic phosphate into close proximity, facilitating their combination to form ATP. It's like a molecular assembly line, churning out ATP molecules to fuel the cell's activities. Once synthesized, ATP is transported out of the mitochondria and into the cytoplasm, where it can be readily utilized for various cellular processes.